Hello, good evening, good afternoon, good morning, wherever you are, everybody, and welcome to our latest OpenFlow cytometry webinar. We're going to be following on from what we did last time, talking about panel design. I'll leave that splash screen there for a second, just to remind you that there are several ways of getting in touch with us during the course of this webinar. Actually, the best way is to use the Q&A function. If you've got any questions, please put them into there and we will answer them hopefully as we go along. But if we don't manage to answer them as we're going, we will make sure that we cover everything at the end there. If you've got any issues with sound or vision or anything like that, we can use the chat function for that. So what we're going to be doing today, as I said, is following on a little bit from what we did at our last session, where we started to look at panel design, generally in terms of multicolor fluorescence. But today we're going to concentrate more on maybe when things can go wrong, and also looking at sample preparation as well. So who are we? So introduce ourselves. Many of you will have been to these webinars before, but my name is Derek Davis. I am the Science Technology Platform Training Lead at the Francis Crick Institute here in dark and dismal London. And I'm joined by my colleagues, and I'll pass over to Kathy. We'll go in order there.
All right. Thank you very much, Derek. So I'm Kathy Daniels. I'm the Flow Cytometry Core Facility Manager here at the Whitehead Institute in very sunny right now, Cambridge, Massachusetts, even though it's a little bit chilly. And I will pass it along to Rui.
Thank you, Kathy. Yes, welcome everyone. It's always a pleasure to participate in these OpenFlow lectures and seminars. So my name is Rui Gardner. I'm the director of the Flow Core Facility at Memorial Sloan Kettering Cancer Center in sunny New York. And actually today I'm doing it from home, and right in front of me is the World Trade Center. So it's a beautiful sunny day, and looking forward for this session.
Excellent, thank you guys. And Rui and Kathy will be doing the practical parts of today. So today will be a sort of combination of theory. We'll have a few slides, but we will actually do some data analysis. So again, it's a way of hopefully reiterating the way we look at our data, the way we assess the quality of our data, and the way we get some numbers out of our data. So we're going to divide it into several sections today. We're going to look at the influence of buffers that we have to get our cells from the flask or from the patient or from the mouse to the flow cytometer. And generally, we're going to have to do not only some staining, but we may have to use some special buffers or fixation or permeabilization, depending on what our question is. We're also going to look at things that can confound our signals. So things like dead cells and the use of viability dyes and the sort of types of non-specific binding that we can potentially come across. We're going to look in a little bit of detail about how we decide what is positive. Many of you might be familiar with the FMO, the fluorescence -1 control. If you're not, or if you're a little unsure about it, we will have a look at that. And that will help us with looking at the effects of data spread that we see often in flow cytometry, whether it's conventional flow cytometry or full spectrum flow cytometry. And then hopefully at the end, we'll have a few minutes and we'd like to introduce you to the OMIP. And if you don't know what OMIP stands for, hang around and we will enlighten you towards the end of this webinar. So whenever we're designing a flow cytometric panel, and it doesn't really matter whether it's a simple panel or a complex panel, our panel design is going to be a bit of a journey. It's going to start by actually thinking about several things. And one of those things is the biological question that we're going to ask. And that might be something that's relatively simple. It could be, how many of my cells are viable, or how many of my cells are expressing a particular fluorescent protein. Or it could be much more complex. It could be a 10, 15, 20 plus color experiment. We also might like to think whether our experiment is going to be a quick and dirty experiment. It may just be checking the phenotype of a mouse, for example. Or is it going to be something that we're going to be doing over a long period of time? Obviously, if it is, we want to make sure that our results are robust so that we can compare our results across that study. There's going to be some prosaic things. We're going to need to think about, where's our target? Is it extracellular markers? Generally, if it's extracellular, it's a little bit easier than if we're looking for something that's inside the cell. And whether it's inside the cell, it might make a difference whether our target is in the cytoplasm or whether it's in the nucleus. And that might mean that we're going to need some special treatment. Generally, antibodies aren't going to go into whole cells without the fixation or permeabilization steps that we're going to need to put into there. So all of that is going to be thinking about our experimental design. We're also going to think about our antigens or our targets. It's not always about phenotyping. This is something we looked at a little bit last time, because this helps us think about which fluorochromes we might use together. So, for example, are our targets on a lot of cells in our sample, or are they only going to be on a few cells? Are we looking for a rare event? Of course, flow cytometry is very well suited for looking at rare events. We need to think about the target density. Something that's very abundant in the cell is going to be much easier to see than something that's less abundant. And also, again, something we looked at last time, the staining pattern. If we've got obvious negatives and positives, that's generally quite easy to see and to design an experiment. The problem comes, and as we'll see when we look at some of the data, is where we get that sort of emerging marker, or a marker that's modulated when we do something to our cells. Because at the end of this, we're always going to be trying to maximize our resolution. Remember, we talk about signal to noise ratio, and our noise comes from a combination of autofluorescence from our cells, from photons, from other fluorochromes in the system, non-specific binding, dead cells, and so on. So the more we can reduce that background, reduce that noise, the easier it's going to be, hopefully, to see our specific signal. So again, as we looked at last time, if we've got antigens or targets that are weakly expressed, we're probably going to want to use a bright fluorochrome to give our chance the best chance of seeing it. And we also need to think about which markers are expressed on which cell as well. Even in a simple 4-color experiment, I could have some cells that don't express any markers, some that express all 4, and then various combinations of 1, 2, or 3 markers. Every time I've got a marker and therefore a fluorochrome on a cell, it potentially reduces my resolution. So there are both good and bad combinations of fluorochromes out there. And this isn't necessarily an easy or a quick process. And the more complex your experiment, the longer it takes. You may come up with what you think is a reasonable experiment on paper, but then you have to go to the cytometer, test it. If it doesn't work, we come back and we start again as an iteration. And it can take a long time. So just to recap, I suppose, on the idea of fluorochrome brightness, we want to know how bright our fluorochromes are, because again, that helps us with assigning our fluorochromes to our markers. We want to generally put a strong or a bright fluorochrome on a weak marker. And we've looked at the stain index several times, actually, in the course of these webinars. And this is the way we can use to assess the brightness of our fluorochromes. What we would normally do is take something like CD4, run it labeled with every fluorochrome that we can detect on our system, and then derive a number. And that number is this thing called the stain index. And in essence, it's defined as the distance between our positive and our negative peak, but we divide it by the width of the negative peak, and that's because cells are autofluorescent in different areas of the spectrum. And this actually helps us in determining which fluorochromes are brighter, because if I look at these two diagrams here, the distance between the positive and the negatives in both cases is the same, but the resolution in this one is less. There's less space here between the negatives and the positives than there is here. Now that means that this will give me a lower stain index than this one, which is great. That helps me rank my fluorochromes. And much of this is out there, that the information is out there either from manufacturers or from your local facility. So this is from BioLegend on the left, this is from BD on the right. You can see they've both qualitatively assessed brightness, ranging from very bright to dim in this case, or 1, 2, 3, 4 ticks on the BD site. And this helps us then thinking about assigning our fluorochromes. So if I'm doing a 1 or a 2 or a 3 color experiment, I'm probably going to choose some of these very bright dyes. It's going to give me the best signal to noise ratio, hopefully maximize my resolution. Once we get to the 15, 20 colors, then we know we're going to have to use some that are less bright. So then it becomes a sort of a bit of a logic puzzle when you work out which fluorochrome you should attach to which marker. One word of caution, I suppose, with these is that these are very dependent on the machine that they've been run. So your machine may have a different power laser, you may have slightly different optical filters, which might mean it's a bit of like a league table. One of these dyes might leap up a little bit or go down a little bit on your particular machine. In the ideal world as well, we should do this particular experiment for every condition we're likely to come across. So not just unfixed surface staining, but fixing our cells in the various fixatives we're likely to use, using the various buffers that we're likely to use, because all of those can have an effect on our fluorochromes, as we'll see in a second. But it's a good starting point. So there are other things that can affect our resolution as well, our signal to noise ratio, which are going to be things that are going to be involved in your sample prep. So fixation can change this. Fixation can affect both your cells and your fluorochromes. Permeabilization, very many times we're going to have to permeabilize our cells to get our marker in there. And we're going to have to do that with some sort of detergent or permeabilizing agent. And most of the time we'll get away with a fairly standard buffer. People will have their favorite fax buffer or flow buffer, which is generally a buffered PBS, maybe with a little bit of protein, some BSA or some FCS in there. But buffering is quite important. Some dyes change their fluorescence if they're outside a particular pH range, and FITC is a good example of that. And it's useful to be aware of that early on. So fixation, fixation we've touched upon several times during our webinars, and we will do in our next one, in fact, as well. Often we're going to have to fix our cells for many reasons. We might have an infective cell. We may have, we might want to store our cells so we can run them when it's convenient to us. And fixation is going to stop your cells. It's going to stop any physiological processes, so they should remain in stasis until you're ready to run them. But if you're going to fix your cells, you're immediately faced with a choice. What am I going to fix them in? And there are two main types of fixative we're going to come across, either alcohols or aldehydes. By alcohols, we generally mean ethanol, sometimes methanol. And alcohols work by coagulating proteins. They change the shape of proteins, which makes them very good if we're doing DNA analysis, because it removes all those proteins that are normally around DNA. But maybe it can be a bit variable with antigen staining, antibody staining, because most of the antibodies that we use in flow or immunofluorescence are directed against a particular conformation of that protein. And if the conformation's been changed, then our antibody may no longer bind. So that might be something that we might need to check. But if we don't want to change the structure of our protein, we would use an aldehyde fixative, generally formaldehyde. Formaldehyde is a cross-linking fixative. It forms bonds with the lysine residues in proteins, and that locks the structure of your protein in place, which is great if you're doing antibody staining, because it probably means that your antibody will still recognize its epitope. Also great for fluorescent proteins, not so good for DNA. The problem with fixation is it's always going to increase autofluorescence. Our noise is going to increase. So therefore our signal to noise ratio may be affected. So we need to be aware of that. Another practical consideration is we need to be aware of the time that we fix our cells. So with alcohol, fixation is pretty much a one-stop shop. You fix your cells in 70% ethanol normally, and then they're fixed. Their autofluorescence changes a little bit, but then that doesn't really change over time. Whereas formaldehyde, we have to be a little bit more careful, because the longer we keep our cells in formaldehyde, the greater we see the increase in autofluorescence. So if I'm fixing in formaldehyde, I would normally do that for a short period, anywhere between, say, 10 minutes and an hour, wash the formaldehyde off, and then store my cells in PBS. Now, alcohol is a good fixative. It's also a good permeabilizing reagent. It dissolves lipids. So that means that you, excuse me, punch big holes in the plasma membrane and the nuclear membrane. Formaldehyde is a reasonable fixative, but it's not a very good permeabilizing reagent. So if I'm looking for an intracellular target and I've fixed my cells in formaldehyde, then I'm going to need to add a permeabilization step. And I'm going to use detergent, but which detergent I use may well depend on my biological question. Where is my target? So if I'm looking for something that's in the nucleus, I need to be quite harsh. And I would use something like Triton. Now, those of you who do Western blots will know that we use Triton to pop your cells to release the protein content. If we use the right concentration of Triton, we can keep our cells intact and we can permeabilize both the plasma and the nuclear membrane. If I'm looking for something in the cytoplasm, I don't have to be so harsh. And in most cases, we want to try to be as gentle with our cells as possible. So if I'm looking for something in the cytoplasm, I might use saponin, or in this example over here, digitonin, which permeabilizes the plasma membrane so we can stain our intracellular markers, whereas Triton permeabilizes both the plasma membrane and the nuclear membrane. So most of the time when we're looking for intracellular markers, we're going to use a fix and perm kit, and you can buy these from many companies, and they're generally going to be, well, they are a combination of a fixative, formaldehyde of some description, and then a detergent of some description. Often we don't know what exactly they are. But we have to be a little bit careful again, because we might have to spend a bit of time optimizing both the time of fixation and permeabilization, the concentrations that we use. So again, this is why a complex experiment can take quite a long time to work up. There may be some variables that we have to work out before we get there. And the reason I'm saying all this is because not all fluorochromes are equal either, and our fluorochromes can potentially be affected by what we do to our cells. One of the things that I tend to do when we train people in cytometry is actually start with fluorescence, and we talk about the chemistry of the dyes. This becomes quite important to know this, especially now in the last few years when we've seen a big proliferation in the number of and types of dye that we use in cytometry. Just to put a few examples here, we won't overload you, but fluorescein. So fluorescein's been around for 100 years or so or more, and it's a small molecule. Now, if I've got a small molecule, that makes me think, one, it's quite easy to get a molecule like fluorescein into a cell because it's not very big. It means that if I've got an antibody and I want to add fluorescein to it, I can probably add not just one, but maybe 6, 8, 10 molecules of fluorescein to my antibody. And small molecules tend to be more resistant to fixation. There's less of them to be interfered with the fixative. Unlike something like PE or GFP, which are proteins, they're big proteins. Now, if I treat those big proteins with a fixative, fixatives are going to change the chemical makeup of proteins, alcohol in particular. If I fix GFP cells, for example, in alcohol, very often I lose the fluorescence. So GFP fluoresces because there's half a dozen amino acids in the middle of this structure here that are very in close proximity. Remember, alcohol changes the structure of the protein. So if we change the structure of the protein, it means that the protein may no longer be fluorescent. It's green non-fluorescent protein, which is why you can buy GFP or anti-GFP antibodies for exactly this case. But if you want to keep that GFP fluorescence intact, this is where I would normally use formaldehyde, which locks that structure in place and keeps the fluorescence. Anything that affects PE will also affect any of the tandem dyes as well. Remember, the tandem dyes are a combination of the donor, PE in this case, and Cy7, which is the acceptor dye in this PE Cy7 example here. Because those two dyes are very close together, anything that changes the structure so they're further apart can change the fluorescence intensity. So sometimes we can get away with staining our cells and fixing them. Other times we can fix our cells first and then do the staining. It may depend on the marker that you're looking for. Again, it may mean that you have to do a little bit of experimentation. So most of these dyes have been around for a long time. Quantum dots have been around for a while as well. One of the newer sets of dyes out there are the polymer dyes, we've talked about, and you're probably familiar with the brilliant dyes, brilliant violet, brilliant ultraviolet dyes. And these are a string of cyclic rings, big long line of these cyclic rings. And rather than each of those cyclic rings emitting light, they transfer energy to a fluorochrome by the process of fret. And it's that fluorochrome that lights up. Now we get a problem with these dyes, because if we're using more than one of those dyes together, because they are very string-like, it's possible that these dyes can actually interact with each other, get tangled up really, just like bits of string, in fact. And we sort of know this from the early days of these dyes, when we started thinking, great, I can put 3 or 4 or more brilliant violet dyes in my panel. There can be a bit of a problem. So in this example, we're looking at CD4, CD8, mutually exclusive markers, or CD4, CD19, T cells and B cells, again, mutually exclusive. But if we look at these two plots at the top, this dotted line represents the line of compensation, if you like. This population, this CD4, obviously shouldn't be positive for CD8, but it looks like it is. And that's because we're getting a little bit of interaction between these dyes, these fluorochromes. So some of the CD8 fluorochrome, for example, is getting tangled up with the CD4 one. We're seeing something that looks like it's double positive. Now we know this, so we can add what we call a brilliant stain buffer. Different manufacturers will market this and will sell you this. Bit of a proprietary formula, but what it means is that you don't get that entanglement, and if we use that buffer, then we get exactly what we expect to see. So this is what we would call something like dye-mediated binding. And sometimes when we see issues with our staining, we have to think about the biology and what we're actually doing to our cells, or what our cells are expressing. We'll show you an example of the brilliant violet interactions in a second when we move over to Kathy. I just wanted a couple more slides to talk about some of the other potential issues that we can get with non-specific binding. And many of you will be familiar with this FC receptor binding, particularly if you're doing blood panels. A lot of blood cells, particularly monocytes and B cells, actually express these FC receptors. So part of their job is to detect antibodies. And that's what these are on the surface of the cell. So, if we're in an ideal world, what we're doing is we're staining our cells with an antibody against our specific marker, which binds, which is great. That's exactly what we want. But in the real world, we also get some non-specific binding. That antigen binding site of our antibody may bind to a different motif on a different protein, or it may get entrapped somehow in the cell. So we'll have a little bit of a level of non-specific binding. But if I'm using a whole immunoglobulin, I get specific binding. It's not what we want, but it's real binding. It's the binding of the FC receptor to the FC portion of the antibody. And I think we talked before about the use of isotype controls in flow cytometry, and I think none of us here on this call would probably recommend them in a panel, but they are useful in panel development because they can help you understand if your cells are sticky, if they are somehow expressing these FC receptors, because some cells express them when you may not expect them to. And if you know you're getting some FC binding, then you can do something about it. And we can do that by blocking, either with serum of the organism that we've raised the antibody in, or more commonly, a commercial FC block, which is normally a combination of CD16, CD32, CD64. Quite easy to remember. There's a nice progression with those numbers. And that FC block contains unlabeled antibodies. So they're going to bind to the FC receptors and block any FC binding of your antibody. The other way, of course, is to not use the whole immunoglobulin, but just to use the Fab fragments. And many, but not all antibodies are available in that format. And basically what we've done here is use an enzyme to get rid of this portion of the antibody. So we've just got the antigenic binding site and the light chains. Or what Miltenyi have done recently is produce these REA, recombinant engineered antibodies, where they've genetically altered the FC portion of the antibody so that it no longer binds to the FC receptor. And it doesn't matter which way you do that in practice, what we would do is put our blocking step onto our cells for a few minutes, room temperature, and then importantly, don't wash them before you add your antibody. And one final thing that we can potentially get is binding of the fluorochromes themselves to cells, not the antibody that they're attached to, but the actual fluorochrome. And there were a few reports of this a few years ago, particularly of the cyanin dyes, and we use those a lot, don't we, in flow cytometry? P-Cy5, P-Cy5.5, P-Cy7, that acceptor part is a cyanin dye. And it was found that the cyanin dye binds specifically to the FC gamma receptor, CD64 receptor. So again, we can do something about this, and particularly BioLegend produced this thing called a true stain buffer. And if you don't use that, what you see here in red is a huge shift in the intensity, almost a log difference in the intensity. That's just the fluorochrome binding to the cells. But binding to the FC gamma receptors. But once we block that with the true stain, then we don't see any non-specific binding at all. And the process is very similar. And remember, that's what we always want to reduce, this non-specific binding. That's a contributor to the noise in the system. So the more we can do to reduce that, the better. Okay, so I talked for long enough. Let's actually go to look at some real data. So I'm going to hand you over to Kathy now, and we'll look at an example of the brilliant staining buffer. So I'll stop my share so that Kathy can take over.
Wonderful. Thank you so much, Derek. And can everyone see my screen okay?
Yep.
Okay. Perfect. Thank you. All right. So as Derek mentioned, the use of a brilliant stain buffer is really going to be critical when you're designing a panel that has more than one of those polymer dyes, right? So I have a data set here that I'm going to thank Anna Longini for, who's at Memorial Sloan Kettering Cancer Center. And I'm going to bring that data set into FlowJo. Okay, I'll minimize this folder. And what I'd like to do is I'd like to show you what happens when we stain in our normal fax buffer or our normal max buffer, and compare that to what the data looks like when we actually use the correct staining buffer. So if we open up and we start looking at our data files from when we stained in a regular buffer that didn't account for this polymer interaction, let's see what it looks like. So we're first going to start off with looking at our CD45 versus our viability dye. Oftentimes when you're analyzing data and when you're looking at it, people tend to start off just looking at scatter alone, right? And setting a gate based on scatter. I typically like to play around with that a little bit and kind of adjust my parameters and maybe start off with the fluorochromes of interest and the markers of interest to see if we can really hone in on the populations that we're looking for. So with that being said, I'm looking at CD45 versus the live dead, and I'm going to optimize my display here through the transform button in FlowJo. So this is not only panel design, but you get some tips and tricks in FlowJo if you weren't aware of them. So we could adjust that with basis and look at the linear log display in a little bit of a better fashion to better pull out our populations. From here, I'm going to set a polygon gate on where roughly where we expect our CD45 positive live cells to be. So it's the negative population for a viability dye and positive for CD45. And we'll call those our live CD45 positive cells. Okay. Adjust that gate a little bit. And from there, let's look at our scatter profile. So we'll set forward scatter area versus side scatter area. And from there, let's set a polygon gate and be very inclusive. We don't necessarily know what populations we're looking for, and we don't want to be restrictive by scatter in order to identify populations. You have a rough idea of where your cell types are, but remember that activation or different treatments that you're exposing the cells to can actually adjust that scatter profile and how the cells will present themselves by scatter. Okay. So that's our scatter profile right here. I'm just cutting out some of this debris. I'm going to double click on that population, and then we're going to look at our single cells. So when looking at our single cells, we're going to plot side scatter width versus side scatter area. And you can see a really nice distribution of aggregates right there and our single cells right here. Put in another gate. And remember, I think Marie mentioned this in the chat, but if anyone has any questions as we're going along through this session or any of our sessions, feel free to use the Q&A and we can address that as we're going along. And I see Derek, which is a good sign. We might have a question.
I'm going to ask you a question.
Okay.
And why have you chosen that particular combination of markers to pick out the doublets?
Sure. So essentially what I typically tend to do is I usually like looking at every combination of the data. So I'll look at forward scatter height, forward scatter area, forward scatter width, and I look to see where I get the best resolution of my populations. There's no one defined way to say it needs to be this way, it needs to be the other way, or you need to look at a specific combination. In looking at this data earlier, I like the way this resolves our single cells. So because it takes a little bit longer for those aggregates of cells to get through the laser, the voltage pulse that's generated has a exaggerated width, and that's why we're able to see those aggregates right there. But I could very easily look at side scatter area versus side scatter height, see a similar population right there, and those are our aggregates. I could take a look at forward scatter width and forward scatter height, so on and so forth. But it's just my preference for today.
Yeah, and I think we might see different ways of doing it later on as well. And I think also sometimes it depends on the machine that you're using.
Yes.
You're getting good resolution there, like you say, on the, this is presumably run on a BD machine or something like that. Because width isn't always a real parameter, is it? Sometimes it's a calculated parameter. So
yes.
Sometimes we use width, sometimes we don't. It depends which machine we're using.
Yeah, absolutely. Like, for example, I believe the Cytoflex, you only have the capability of looking at width off one detector. So you're not going to have the options like you have here to play around so much. So it really is absolutely dependent on the instrument that you're working with and the cells and how they resolve on that instrumentation.
I think the important thing that we'd always say is it doesn't matter which way you do it, it's important to do it, because otherwise you're running the risk of not doing single cell analysis anymore.
Exactly. And remembering that a negative cell and a positive cell stuck together is going to look like a dim positive, right? So we might be looking at our data and not realizing that we're kind of falsely generating a population that's not real. Okay. All right. Thank you, Derek. It's a great question. A lot of people have pretty often. So from there, what I'm going to do is I'm going to go ahead and I'm going to start looking at some fluor combinations. So this specific combination of fluors is very heavy in the brilliant violet and brilliant UV dyes. So what I'm going to do first is I'm actually going to go ahead and I'm going to take a look at our MHC class 2, and I'm going to take a look as well at our Li6C. Okay. And we can see here, this is what we expect to see from data that resolves pretty well, right? We're able to see populations that are pulling out very well. And this is just our regular max buffer, right? And we have a brilliant violet 650 versus a brilliant violet 711. If I come in, I'm able to easily identify these populations for our MHC class 2 positive, right? So if I wanted to come in here, I can set this gate and I can be confident that that's a single positive population. Okay. So I can go ahead and do any gating that I want. Now, this might not be relevant to your gating and what you're looking to do. I'm really looking at the staining itself and how these populations resolve, and not necessarily the immunology behind what these markers and what we're looking to pull out. Now, if I take that same data set, I'm going to remove these gates because they're not really necessary, and I swap it up, I want to look at a different fluor combination. What I'm going to do is, instead of the BV650, let's look at another one of these markers. Let's look at CD11C. Now, right away, I think we can all look at this and see that there's quite a difference here in how the populations are pulling out, right? Let me go ahead and customize this axis and adjust the width basis a little bit. you can get that full distribution. We see a bit of the spread from that BV605 into the BV711. So we see that distribution that we expect to see when there's high spread. But what we also see is this population right here. When we're looking at this population, it's, we're able to resolve it, but it doesn't look like what we expect flow cytometry data to look like that's been compensated or unmixed correctly. It looks like there's a curving of the population, which is what we expect to see if data is undercompensated, right? The reason for that, if we're thinking about compensation, is, let me do this. We expect the MFI of the negative and the positive to match in the spillover detector, right? So if this is our negative population right here, and this theoretically would be our single positive for this BV711 Ly6C, but the population has completely shifted over, okay, in a manner that we would not expect. When we see it done correctly, we see that the MFI of the positive and the negative tend to match up in that spillover detector, right? So BV711 right here. So this was a panel that was put together and stained with a regular max buffer. What I'm going to do now is I'm going to do a very similar analysis on a sample that was stained with the Brilliant buffer, taking into account the fact that we had multiple polymer dyes in the panel. So I'm going to go ahead and I'm going to open up this data file. I'm going to close any of the plots that we don't need. And I'm going to look at our CD45 versus our live dead. Okay. We can see that's resolving very well. I can make any adjustments if I need to, but it's really the same sample that was split up. So it should be pretty much the same, which it is. We see our scatter exactly the same, right? Or as close as possible as it could be. And then we look at our single cell population. And if I open that up, it automatically is showing us the CD11C versus Ly6C. Now, what do we see? With the Brilliant stain buffer that takes into account how those polymer dyes can interact with each other, we no longer see that swooping of the population, and we don't see the false positive of that CD11C BV605. Now you can imagine without using that Brilliant stain buffer, you could think that there was a population that was dimly positive for CD11C and also positive for Ly6C. But once we account for the interactions of those dyes, what we now see is that the MFI of the positive and negative, and sorry for my little tilty line there, really matches over in the spillover channel. So you can imagine what can happen as you start to add more and more of these polymer dyes to your panels. I'd also like to note something very important that a lot of people don't realize, which is that a lot of these polymer dyes are actually tandems. So you have to be very careful with how you treat tandem dyes, because typically the BV421 or the BV395, those are the base dyes, and there can be tandems, and that's where a lot of the longer shifted wavelength emitting dyes come from in the Brilliance. Another thing that actually just popped up on the Purdue list was actually about staining volumes, because oftentimes people are utilizing these Brilliant dyes in their panels, right? And they might have a 20 or 25 fluorochrome panel that they're looking to work with, and volume becomes something that you have to think about. So in addition to the Brilliant stain buffer or the super bright buffer, they actually have Brilliant stain buffer plus, or a super version of it, where you could actually have a more concentrated version of this reagent, and you have to add a smaller amount to your well or to your tube so that you can actually have a little bit of a better condition as far as volumes are going or concerned when you're staining. So this is a very beautiful example of two samples, or the same sample stained with the same exact set of fluorochromes, and how the fluorochrome, or the choice of using that brilliant stain buffer, really helps you out. And before I pass it back along to Derek, I'm just going to show an overlay of those two populations, or of those two FCS files rather, to show the difference. Okay. If I open that up, I'm going to use some larger dots that it can help show our point a bit better. It's a little bit messy when we look at it this way, but I think the important thing to note here that some people might have some questions about is, fluorochrome brightness, and, is one brighter than the other? Now, these two are the same. And what's important to also note is that if it was brighter in one than it was the other, for that BV711, it would actually show a pattern of overcompensation. So that's not something we'd expect anyway. And I see Rui now, so I'm looking forward to another question that hopefully came through that Rui has, that I'll do my best to answer.
Actually, it's more of a comment that, recently I also saw, I forget now in which forum, but there was a discussion about the brilliant stain buffer, and somebody was mentioning that, I have a panel with, I don't know, it was 3 or 4 BV dyes. And of course, most of them were tandems, as you mentioned. And that person was mentioning that, I've never had any problems, never used the brilliant stain buffer, and it worked perfectly well. And, good for that person. I'm really happy. But, it may work, but most of the times it won't work. So you may be lucky and you don't see any interactions, or maybe just the populations that were being looked at were kind of mutually exclusive, and it's just lucky that no interaction took place. But in general, they will take place, and so it's really important to use this buffer. So I just wanted to comment that yeah, you might not see problems sometimes, but in general, you will see issues, and sometimes they may be so subtle that you'll have a hard time in understanding if it's a biological phenomena that you're seeing, or if it's just an artifact, right? And so that's why it's so important to just apply best practices and use that staining buffer.
Yeah,
I just wanted to comment.
Absolutely, and that's a great point, because if we look at the CD11B versus, if you look at, BUV395 and BB711, let's focus on the fluorochromes and not the markers. If we look at that, we don't really see that issue, right? So depending on how you're looking at your data, it could really change, right? Whether or not you're identifying those problems. But if we just went ahead and looked at, not my favorite, but a histogram, and then we looked at this, you might, it's probably not the best, but you're seeing this tail, and there can be, or whatever the marker is that you're seeing that spread into is actually, it was the BV605. You might have this population that you're assuming is BV605 intermediate, but it's the spillover from that other detector, and you're making a false assumption. And that's why you always want to go in and you actually want to look at the N by N data and actually do a quality check, not just by time for fluidic stability and everything like that, but you want to check the panel as a whole to make sure that you're seeing the populations without any major indications of issues due to fluorochrome combinations that aren't working out so well, staining that hasn't been optimized, or single color controls that haven't been optimized as well. Okay. So that's my example of the brilliant stain buffer. And with that, I'm going to stop my screen share and pass it along to Derek.
So we've done that. So let's think about what else can confound our analysis, but again, particularly thinking about the noise. And one thing that we always recommend that you do is to identify the dead cells in your sample. I mean, sometimes that's the end readout of your assay, but particularly in immunophenotyping, we want to make sure that we remove those dead cells, because no matter how good our processing is, we're always going to have a few dead cells in there. And once cells die, several things happen. First of all, the scatter changes, something that, again, we've looked at before. We've seen, I'm sure you've seen this as well, particularly if you deal with suspension cell lines. This is a Jurkat cell line. Now I can be pretty sure from experience that this is my live cell population mainly, and this is mainly dead cells. And I know that those are the dead cells because when a cell dies, it becomes permeable. And when it becomes permeable, it takes on fluid from the outside. It becomes osmotically compromised. And actually it gets bigger, it swells. But what that means is the refractive index between the inside and the outside of the cell is less. So as our laser passes through, we don't get as much refraction. We get not as much light being bent. So our forward scatter signal actually goes down as cells die. At the same time, those cells are starting to get a bit blebby. We get an increase in our side scatter or a 90-degree light scatter. So our cells, as they die, move from sort of here up and maybe back down this way as they start to break up. Now, if I've got a relatively nice round cell that grows in suspension, sometimes scatter is a good way of getting a good handle on how many cells are dead. It's not the best way of doing it, but it gives you a pointer. If I'm looking at cell line, I'd have a much harder job doing that. And we'll show you in a second how we can specifically identify those dead cells. The other problem we get with dead cells is that their metabolism changes and their autofluorescence changes as well. And if I put a region around this population and around this population, and here display the APC channel, you can see that my live cells in red have a lower autofluorescence than my dead cells in blue, because those dead cells have changed. Now, if I were looking for something that were weakly positive in this APC channel, those dead cells might fool me. I might be thinking, oh, there are some positive cells there. It's not, because they're dead and they've changed their autofluorescence. And we're going to use the fact that, I suppose, our cytometric definition of cell death is that our cells become permeable, their membranes become compromised. So knowing that, we can use some knowledge and we can use some fluorochromes to help us remove our dead cells. And for many years, the way we did this was by using DNA binding dyes, things like propylium iodide or DAPI or any of those other dyes that you're probably familiar with that bind to DNA. The principle, as I said, is the fact that a dead cell is permeable. So if I add propylium iodide to a culture or a tube of live cells, that dye is not going to go into the live cells. Okay? Can't get in, or it gets pumped out very quickly. With a dead cell, compromised membrane, that dye is going to get in, it's going to make its way to the nucleus, bind to the DNA, and it's going to become fluorescent. So when I run those on the cytometer, I can see my cells that are dead and have a relatively bright signal for propylium iodide, and my cells that are alive that have a low signal for propylium iodide. Now that's great if I've got unfixed cells. But if I were to fix these cells, and remember fixation also permeabilizes cells, if I were to fix these cells and then do that assay, everything would be positive for the DNA binding dye. I wouldn't be able to tell the difference between the ones that were live before I fixed and the ones that were dead before I fixed. And often that's exactly what we want to do. If we're looking at phosphosignaling, for example, where we're looking at cytoplasmic markers, and we might only see a small shift in fluorescence in positivity, I want to know that that small shift is real. So how do I do this in dead cells? Well, we use the same process, the fact that our dead cells are permeable, but rather than using a DNA binding dye, we use a protein binding dye or an amine reactive dye or a live dead dye. I'll get a little bit of staining on my live cells because obviously they have a few proteins on the surface. With my dead cells, compromised membrane, that dye is going to get in there and it's going to bind to all of the proteins inside my cell. So now when I run them on the flow cytometer, my dead cells are brightly positive, my live cells less so. And the great thing about these dyes is that I can now fix this. As long as I do the staining before I fix it, I can fix in ethanol or I can fix in formaldehyde, and that staining gets locked in place. So that means that even after fixation, I know which cells were dead before I fixed and which cells were live before I fixed. And the great thing about both of these, both the DNA binding dyes and the protein binding dyes, is that they come in a variety of different flavors, so different excitations, different emissions. So in the top row here, we have 3 different DNA binding dyes, DAPI, propylium iodide, and TOPRO3. And you can see that they pick up the dead cells. We get pretty much the same percentage in each. on a different day. So this is why the numbers aren't quite the same. So, but the row below, we've used 3 of the different protein binding dyes. So I show these because they're all excited by different wavelengths. So UV, blue or yellow in the case of propylidiamide, and red in the case of topro, or UV violet and blue in the case of these live dead dyes. But again, you can see it doesn't matter which of these protein binding dyes I use, I get pretty much the same percentage. So that's great in terms of panel design, because it means that I'm nearly always going to have a channel that I can fit one of these dyes in. And I don't have to worry too much about signal to noise ratio, because whether I'm using a DNA binding dye or a protein binding dye, I'm going to get a lot of that bound. So I'm nearly always going to get a very good signal to noise ratio. And also, when I do my analysis, the first thing I'm going to do is remove these dead cells. So those dyes, DNA binding dye or protein binding dye, are not actually going to interfere with any of my other fluorochromes. The cells that I'm interested in are never going to have those dyes on them. So hopefully that's actually already familiar to a lot of you. So I'm going to hand you back to Kathy again. She's had a very short coffee break there. And we'll look at a couple of examples of this.
Thank you so much, Derek. And I actually should have brought more coffee so I could have had a longer coffee break or more coffee-ish coffee break. Okay, so talking about viability dyes is something that I think myself and many other cytometrists are pretty passionate about for the reasons that Derek mentioned. So what I'm going to do is I'm going to walk you through a file and an analysis of immunophenotyping panel from Toby Oni here at the Whitehead Institute, who we're very thankful he allowed us to use his data file. So if we open this up, this is from a SciTech Aurora. What I'm going to do first is I'm going to analyze the data as if we did not have a viability dye, and not really understanding where our live and dead cells might be. So Derek showed some data before about the scatter profile and how it can shift due to the changes in morphology of the cells. But if you're starting out, you're not really sure, right? More often than not, what you're going to do is you're going to try and go ahead and cut out some of the small debris, and any of these larger populations you're going to include in your analysis. So if an investigator comes in and does that, right, and they say, okay, this is my scatter profile. Again, I'm keeping it open because I'm looking for all sorts of different cell types. And from there, what we're going to do is we're going to go ahead, move this. Sorry about this. There's just a panel for Zoom. From there, what I'm going to do is I'm going to go ahead and look at our single cells. So again, we can look at side scatter width, side scatter area. Because this is the Aurora, there's side scatter off the violet and also side scatter off the blue. And that's why you see SSCBA here. That was side scatter blue. And what I'm going to do is I'm going to go ahead and look at our single cells right here. From this, I'm interested today in this panel in looking at our CD3 positive cells. So we're going to go ahead and look at our Alexa Fluor 488 CD3. And for now, it's resolving pretty well. So I'll leave it versus side scatter. There's definitely, a benefit to plotting it versus another fluorescent detector, especially if you have a hard time resolving populations, or if you're working with cells that have a high autofluorescence. Because I'm able to resolve this pretty well here, I'm just going to go ahead and set a gate on where I have my CD3 positive population that's resolving. And these are my CD3 positive cells. Okay. So we have about 35.2% CD3 positivity. And from there, what I'm really curious about looking at in this specific panel is my CD25 positive CD4 T cells. Okay. So what I'm going to do is I'm going to plot my CD4 versus my CD25, okay? So we can see this data is not looking so great right here. We have a little bit of a extreme negative going on, right? So that's something we're gonna explore in a little bit. For now, I'm gonna try and optimize this with basis a little bit to see if we can pull out these populations a little bit better to pull out my CD25 positive CD4 T cells, indicating that there's some type of activation of these cells. Okay, so when I'm looking at this, I'm going to set a gate right here. Okay, so I'm going to call those my activated CD4 positive T cells. And we have a population that's about 6.05%. Okay, and that's of the parent. Now, what I'm going to do is I'm going to take the same data file and I'm going to look at it with the inclusion of the viability dye. So I'm going to, if I open up that same FCS file, you can see that the same, or that nothing opened up, and that's because this is the first gate that we put on this file. So what I'm going to do is I'm actually just going to change this to look at DAPI. Okay, and we're going to see if we can customize this axis and improve. Okay, and we see right there, we have our live cells that are negative for DAPI, and we have our dead cells right over here. I'm going to go ahead and I'm going to have my first gate now not be on scatter, but instead I'm going to have that be on this viability dye. So I'm going to call those our live cells. So we have about 80.9%, not too bad. And then from there, what I'm going to do to not bring in bias from how I might data sample differently from a minute or two ago, I'm going to go ahead and I'm going to take that scatter single cell CD3 and activated T cell gate, and I'm going to copy it from the analysis that I just did. And I'm going to paste it here onto this live cell gate that I just made. We're going to make sure everything looks okay. So we're going to go to forward scatter and side scatter blue. And what we see here is this pattern actually looks pretty different, right? So we do have a difference in where we see our cells that are live. So before, if we looked at forward scatter versus side scatter, you can see that there's definitely this population right here that seems to be a majority of where the dead cells are. But there are some cells that were DAPI negative that were still within the scatter gate, and we want to include those cells in our analysis. Okay. So from there, we look at our single cells. That looks good. We see our CD3 positive cells. Now we actually see this nice difference here where before we only had 35.2% CD3 positive cells, and here we have 38.8. So we're getting a better representation of our CD3 positive cells. And what I'd also like to note is that it might be a little hard to tell, but there are a little bit more potentially, without doing a deep analysis, cells that are showing up higher in side scatter here for the CD3 positive when we haven't excluded our dead cells. Okay. From there, I'm going to double click on that population. And here, when we're looking at our CD25 positive, CD4 positive T cells, what do we see? We see a pretty dramatic difference across these files. Okay, so actually it's the same file rather, so I misspoke a bit, but we see a very dramatic difference in when we exclude the dead cells with a 3.53% positive population, as opposed to when we don't include the viability dye in our analysis, where we have almost double the amount of these activated CD4 positive T cells. The other thing that's very important when we're looking at this to keep in mind is that you also see this CD25 positive, CD4 negative population. Okay, so this population completely disappears once we include the viability dye. The other thing that's important to note is that it cleans our data up quite a bit. So if I adjust that width basis back to where it was before, you see kind of all of that, all of these events down here that were a little bit more in the way of the extreme negatives, right? And we're seeing kind of some dirtiness in our data. Once we've cleaned that up through excluding those dead cells, this data file looks much, or this same data file looks much cleaner because we've removed those dead cells. Okay. So you can imagine as you're going through and you're doing your analysis and you're looking for very small changes in population expression, how critical it really is to include a viability dye. Now, some people might say, all right, Kathy, well, you know what? I don't have room in my panel for a viability dye, and I can just do that gating based on scatter. I've been doing flow cytometry for a long time. I know where the scatter gate should fall for my live cells and also for my dead cells. So let's take a look at that, right? Using this same file, we have our DAPI negative live cells right here. And then I'm just going to create a rough gate for my dead cells. Okay, so we have our dead cells and we have our live cells. When we open up the layout editor, what I'm going to do is I'm going to take these dead cells and drag them in. And I'm going to double click on this plot right here. And I'm going to change the x-axis to forward scatter area. And the y-axis, I'm going to keep at the side scatter off the blue. Okay. What we can see is there are a lot of cells right here in this lower left-hand side of the axis. But then we also see quite a bit of events that are right where we expect our live cells to be. So let's do an overlay of that and look at those live cells. And what you can see is when we look at the live cells, which are in blue, and the dead cells that are in red, there's no way for us to pull out 100% of what our live cells are if we were to just put a live cell or a scatter gate on our population. Because we have cells that are in this gate that are all over the place as far as scatter goes. So if you are working with rare populations, if you're working, trying to figure out a really reliable data set to understand where your cells are as far as population expression and intensity, all of that, you can't really rely on data that you're just using a scatter gate on for viability. It's something that we hear fairly often in SRLs, and it's something that we try and combat quite a bit because it is so critical and because it will have such an impact on your research. Okay, so two things. We can have altered population percentages, and we can have a little bit of dirtiness in the data. And also, we can't just rely on scatter for our viability gating. Okay, so with that, that's my presentation on viability dyes and why they're so important. If we have a little bit of time later, I might discuss the impact of utilizing viability dyes and looking at fluorescent proteins as well, but we'll see if we have a little bit of time for that later. And were there any questions that came through, Derek?
Not that we need to do specifically for this bit.
Okay, great. Thanks, Derek. Stop my share.
Stop my share. Dropped out of PowerPoint. There we go. Right. So the last thing that we want to look at practically today, where we can bring Rui into the equation, is to look at the effect of the data spread that we get when we're looking at multiple fluorochromes. So we looked in a previous webinar about compensation. We know how to compensate. We know this is PE. So I've got some PE cells here, some PE negative cells, and we're spilling over into the per-CP channel. And we know that we're correctly compensated here when, as Kathy was demonstrating earlier, when the median of the positive population is in the same place as the median of the negative population. But what we see in many dye combinations is this data spread, okay, which is a combination of the spillover, but also the counting error that we make in our flow cytometer. And we're bringing this population from the high end of the log scale down to the low end of the log scale. So we're revealing the error that we make. And this has a big impact in our experiments because, you know, if I put my cursor there, just above that negative population, if I was looking for a population that's weakly per-CP positive, but PE negative, I've got a reasonable chance of being able to see it. It's not very high above the background, but I could probably see it. But if that population was also positive for PE, then I probably wouldn't see it. I wouldn't see that as positive. It's going to be consumed by that data spread. And this is something that becomes very important in multicolor experiments, because I want to know how I'm going to define something that's positive in that per CP channel, or in that double positive channel. And this is where the very important fluorescence -1, or FMO, control comes in. So we're all familiar with our single color compensation controls, aren't we? And they're great because they help us account for the overlap, and they're very easy to understand. We're looking at photons from a particular fluorochrome that end up in different channels, and then we account for that. The FMO is the inverse of that. We're looking at photons that end up in a particular channel that we're interested in that aren't actually from the fluorochrome that we want. So let's take this relatively simple but poorly executed experiment where we're looking at 4 fluorochromes excited by the blue laser, FITC, PE, PSci5, and PSci7. Now I know that in my PE channel, that's where I'm going to get the fluorescence from my PE labeled marker, whatever that is. But I also know that if I've got those other fluorochromes in the system, some photons from FITC will end up in the PE detector, some from PSci5 will be in the PE detector, and some from PSci7 will be in the PE detector. Now I can account for those by the process of compensation or unmixing, but I want to know when I run that fully stained sample, how many of those photons have come from the actual PE marker that I wanted to use. And that's where the FMO comes in. So the FMO is a sample that's been stained with FITC, PSci5, and PSci7, but not the PE. So now I know the background level. Again, it's noise, isn't it? It's photons from unwanted fluorochromes that are ending up in that detector. And if I do that in a more cartoon form, these are several of the controls we're going to run in any experiment. We're going to have an unstained sample, gives us the background level in this is our PE detector, or a tube, or a rain catcher, however you prefer to look at it. That's our true unstained background noise. We're going to run a single color control, which one will show me that I'm getting staining in the PE channel, but that's not the importance of the single color control. It's to determine where the photons from that PE have ended up. But my FMO is going to contain, in this case, the FITC, the PSci5, and the PSci7 markers. And we know that some photons from each of those fluorochromes ends up in the PE detector. So then when I come with my fully stained sample, I know if I see any increase, that must be due to the PE alone. Now, well, as we'll see in a second, we don't always need FMOs for every color. If I've got an obvious positive and negative, I can get away with it. The importance of this control is where we're getting a change or a gradation in the fluorescence that we're seeing from our markers. But let's see this in practice. So actually, I'm going to hand over to Rui this time, and we'll look at an example of how the FMO is extremely important to us.
Yes, thank you, Derek. As Derek was mentioning, the FMO controls are extremely important and used for gating, right? So if we want to know exactly where we want to set the threshold between negative and positive, we're going to use an FMO, fluorescence -1, for that marker, for that particular marker. And, that FMO is going to account for all of the spillover, all of the photons, as Derek showed in that slide, all of those photons that are entering the detector that we want to use for a given fluorochrome, right? So, if we're not staining with that fluorochrome in particular, any photon that's entering there is going to be from the other fluorochromes, and so we will account for the total background that we're seeing, that spread that he was showing. So that's one of the main uses of the FMO. But in terms of panel design, actually the FMOs can be extremely important, and a lot of times people will they want to skip of course a lot of the controls, because You may have a precious sample. It may be difficult to, you're spending more money. If you have a 20 color or 30 color and you're doing an FMO for all of the markers, we're talking about 20 or 30 FMOs, right? That could be quite expensive. And actually, if you're thinking of different tissues, you should use an FMO for different tissues, because the FMO has to be the multi-stain sample without one of the markers, right? Without stained without one of the markers. And so, and this is to account for differences in expression in different tissues. So you'll have different spreads, and if you're looking at different tissues. So overall, the FMO is quite useful. But like I was saying, with a 20-30 color panel, people start taking away some of the controls, and actually, the more colors you have, the more important it is to have those controls, because it starts being very difficult to troubleshoot. If you have 2 or 3 markers in your panel, and you have a problem, well, it's relatively easy to just go to the data and identify what the problem is. If you have a large panel, 20, 30 colors, and if you see that you have lack of resolution in one of your markers, then what is the problem? Is it bad compensation controls? Is it a bad fluorochrome choice? Is it just the fact that you didn't account for the fact that there are the 2 fluorochromes that are co-expressed? Or 2 markers that are co-expressed, and so those fluorochromes are overlapping and interacting with each other. So it is important to actually have a series of controls and strategies to be able to troubleshoot that panel. And so we want to show the usefulness of having an FMO control in your panel to be able to troubleshoot. So let's say you build a panel, and let's just show a very simple example here of a 6-color panel, 1, 2, 3, 4, 5, 6. So we have PE, PE size 7, PE size 5, APC size 7, APC, and FITC. Now, we built this panel on purpose in a relatively poor way to show the importance of, so let's say we set up this panel, we run the panel, and now how do we know where the problem is coming from, or if we have problems, right? So just look, for simplification, since Kathy already showed how to, gate and how to get to your, to your populations, I'll spare that, for the sake of time, and just show you. So we gate the lymphocytes. By the way, we didn't use a viability dye here, not because we wanted to skip a viability dye. We don't need to skip a viability dye. We should never skip a viability dye. But these are lyophilized cells, so it wouldn't make sense to use a viability dye in this case. So we didn't use it. But if these were live cells, obviously it would be important to use a viability dye. We use a double discrimination here. It was just with Ford scatter, height versus area. And we identified the CD4 T cells, and by the way, I forgot the objective of this panel, or one of the objectives was to look at regulatory T cells based on CD127 and CD25 expression. Now, maybe some of you are not familiar, but we should see a population standing out here that is negative or very dim for CD127 and very positive for CD25. And as you can see, after gating the CD4 positive cells, we actually just see kind of a blob, right? So something went wrong with this panel. We're not resolving. Of course, it could be with the instrument, but here we're assuming that we've QC'd the instrument is performing well. So we've titrated all our reagents. So imagine this was a 20-color panel. Where would you start even identifying where the problem is coming from? How do you know where your problem is? And so one of the ways to do this is to start by, you can start by looking at, okay, so if I have a problem here, maybe spread has been introduced. If I lost resolution, or if I'm expecting a certain resolution, maybe there's some spread that was introduced by spillover, right? So one of the first things you can do is to add, let me see if I find here where the data is to compare your single stain controls. In this case, they need to be the cells, right, that you're using, the same as your sample. And that's why it's important. And here we're talking about you're still optimizing your panel, by the way. Once you've optimized it, you can reach the conclusion, as Derek was saying, if you have populations that really resolve, you don't need to have an FMO, right? That would be pretty obvious. But here we're trying to troubleshoot the panel and optimize it. So we're going to do an FMO for all of the markers, but we're also going to have a single stain control in the cells so that we can compare the single stain with a multi-stain, so the fully stained sample, and see if there was an introduction of spread in the negative population. And like this, we can start by identifying, oh, I have a lot of spread here. Now I can go back and look at my FMO controls and see if I remove one of the fluorochromes, or one by one, I can see if they have an impact in that spread or not. So that is the strategy. So let me see. So we have, as I was saying, we have 127, which is in PE size 7, and CD25 in PE. So let's start by looking at those two, right? The PE size 7, and so I have here a compensation control. So I'm going to open up the single cell. So I've added the same gating strategy. So I'm going to open up the single cells. Actually, I'm going to do the following, because I want to do an overlay. So I'm going to add it here, and then I'm going to say, okay, I want a histogram, and then here we want to look at, what did I add for PE size 7? So we want to look at PE size 7, right? Let's put this up here, not to confuse. So PE size 7. This is the single stain control. Let's overlay it with the actual sample, right? Let me see. The sample is here, so I'll just add the same gating, and you can see that just by looking at this, that already something is wrong. So the blue, I have my video in the way. So the blue is the multi-stain, and the red is the single stain control. Now, sometimes it may be difficult to see because, to compare this properly, because we have maybe less events for the single stain control. You can do things like, I can come here instead of in the y-axis being automatic. We're actually going to choose, for instance, modal, and this will help us kind of visualize things in a better way and compare them. And you can see that this would be most likely the distribution of the negative control, and then we have positive. It's going to be a continuous of CD127, but the negative is quite tight, right? And actually, I could show you if you'd like. I can even show the unstained, where it's pretty tight. But watch notice the spread of the negative population for the multi-stain control. So we've added more spread. We can't see much here on the right because it's a continuous expression, but on the left, we can see that there was a lot of spread introduced, right? And the spread will go both ways, into the negative and the positive side. And so it's very obvious here that one of the fluorochromes, one or more of the fluorochromes that we added introduced spread into the CD127 and probably affecting this the resolution. Now let's take a look at PE. So I don't know, I actually haven't even looked at PE yet. Let me see. So if we have our single stain control for PE, we can do the same. I would add this, again, do the same histogram. And then here I'm going to choose PE, and then I'm going to add our multi-stain again, which would be this sample here. So single cells, and we're going to add it to our, and so here you go again. Now I would just change again from auto to modal, for example, you could use, and you can see here that, well, comparing both, and again, thinking of the blue as looking at the blue as the multi-stained, you can see that there is some spread that's introduced, but not as much as here, but there's still some spread. So we're actually having an influence from one or more fluorochromes into not only CD127, but also CD25, although I would be much more concerned with a CD127, right? And you could start looking at, well, this is the PE size 7 channel. What if I look at, maybe it's APC size 7, but to be honest, CD8 is not expressed. The cells that we're looking here are CD4 single positive, so they do not express CD8. So APC size 7 is not interfering, right? Because we don't have any APC size 7 on these cells coming from CD8. And you could go on. And again, if you have a 20 or 30 color experiment, it would be pretty hard because, in PE size 7, we could have PE, we could have PE size 5, we could have APC size 7, even FITC could, well, that's a bit hard, but even FITC could influence somehow, depending on how much FITC is being expressed. So it would be kind of hard to actually understand which would be the fluorochrome that's affecting this. So like I said, the best way is to run your FMO control at the same time, and then go systematically one by one and see, as you remove one fluorochrome, see if you get an improvement on this, on your population of interest, right? And again, for the sake of time, because I'd like Derek to also talk a little bit about the OMIPs, but so for the sake of time, we went ahead and we ran an FMO for CD127 PE size 7. Sorry, let me say it again. We ran an FMO for all of them, but I chose to show you the FMO for CD4. That's what I should have said. CD4 PE size 5, right? And this is the FMO, so you see there's no positive CD4s, but we gated whatever was CD4 or CD8 negative, right? And now with this gate, so compared to this gate, we're showing the same population, essentially, although of course we're including these cells also because we don't have CD4, so we're including CD4 positive and CD4 negative cells, but that are negative for CD8. And when you look at this, you realize that, oh, okay, now my population is popping out, right? And this is my population of interest that I wanted to see. So the resolution improved dramatically when we removed PE size 5, right? So now we know that at least PE size 5, and again, for the sake of time, I'm not going to go through all of them, just wanted to show you the power of an FMO. When we remove PE size 5, suddenly, boom, we got the population of interest being resolved. So now we know that PE size 5 is not a good fluorochrome of choice because CD4 is co-expressed with CD25 and CD127. And so PE size 5 is definitely impacting the resolution in PE size 7, and actually, and again, like I said, when I showed here the strategy here, we're already seeing that there's also some impact on PE, and in fact, PE size 5 was also affecting the resolution of this population in the CD25 PE channel. So again, a nice way of if you do this systematically, you're optimizing a panel. So you have your first panel of choice, you run it in the instrument, you run all of the FMOs for all of the parameters, and then you start analyzing your data. If you see that there are populations that you expect to resolve, but they're not being resolved, using an FMO and actually starting by looking at all of the channels that you're using, comparing the single stain with a multi-stain and seeing if there's an impact or not. And then if you do see that there's an impact, you can go and check the FMOs on that channel and see which FMO will actually show an improvement. If they show an improvement, it means that fluorochrome that you didn't add is at least one of the culprits, right? You could have several of them. And so again, this will help you to start identifying which are the fluorochromes that are really not, the best choices. So again, a nice strategy. And like I said, if you do this systematically for all of the channels, you might really be able to troubleshoot this very quickly. And this happened, and this works just the same for full spectrum flow cytometry, right? But instead of talking about channels, we were talking about fluorochromes, right? But you could use exactly the same strategy. So We do suggest to run all of the FMO controls when you're first optimizing your panel. Once you've optimized your panel, there are maybe things like, again, like Derek said, if you have some separation like this, you don't need an FMO to set a gate. I would still use them if you can, but if you have precious sample, or if you just don't want to spend all that money because you have such a big panel, then maybe there are some circumstances that you can remove those FMOs. But for optimization, we would really recommend you to use that. I don't know if there's any question. I just saw one coming here in the Q&A. Is it common practice to group FMOs together in one tube? And do you need to test single versus grouped FMOs when optimizing? First question, no, we don't add the FMOs together in one tube. It may be difficult. I mean, if you have some kind of barcoding, maybe you could, but no, it's common practice to actually have different tubes for the FMOs to simplify, and to avoid any possible interaction between the samples. And then the next question was, do you need to test single versus grouped FMOs when optimizing? So you can actually use even fluorescence -2, fluorescence -3, depending on the complexity of your panel, and to look for these kind of interactions and troubleshoot. So you can be as creative as possible. Nobody should tell you that, no, don't use those, they're completely useless. You can definitely look at different strategies. Again, you can test a single versus a grouped FMO, or actually I would say test single versus FMO. I wouldn't group them particularly. I don't know if Derek or Kathy have any recommendations here, but I would say I would prefer not to group them and just run them, individually. It doesn't take much time, and you can even do this in if you have these capabilities in your instrument with a plate reader. But anyway.
Yes, I probably wouldn't group them either. I think the important thing with FMOs is that you do them. I think reviewers in particular, are much more clued up than they used to be. Mm-hmm. The idea of the FMO, and as we've said several times, you can probably get away without it if you've got that obvious positive and negative. But if I'm doing a big panel, I probably would run them at least once at the beginning, so you've got that data. And then you can say, actually, in my panel, I only need it for these particular markers.
Right, and you never know especially with human samples. You never know how the pattern is going to be from donor to donor or patient to patient, and sometimes something like a CD4 or CD8 that resolves very nicely. Sometimes in a certain patient, the pattern may look completely off, and an FMO there would be ideal. But it's difficult because in this case you would need an FMO for each patient, which will never happen in practice. So there are some caveats to FMOs. I think we mentioned this in other sessions. It's not a perfect control, but it's by far the best control for gating. But for optimizing, I think it's a really nice tool to troubleshoot your panel.
Okay, thanks, Rui. So we've got a couple of minutes left. We just wanted to put a couple of slides in, and I need to get control back of the screen. I can do that because it's my webinar. Right. So we just wanted to, in the last couple of minutes here, I know we're almost out of time. I mentioned at the beginning we were going to talk about the OMIP. Okay, and if you've not heard of the OMIP before, they can be very useful things. And OMIP stands for Optimized Multicolor Immunofluorescence Panel, which is why we always talk about OMIPs rather than saying it all out loud. And these are a specific journal type in the journal Cytometry Part A. And very often people are doing research on the same cell type. It might be Tregs that Rui was just looking at there. It might be tetramer positive cells or something like that. And if everybody is looking for the same sort of cells, it'd be great if we had a maybe unified or a optimized way of looking for those. And that was the rationale of the introduction of the OMIP. And really, so somebody has done the hard work so that you don't have to do that. And each of these OMIPs has the same format. And ideally, like any paper should be, it should give you all the information that you would need to repeat and run a similar experiment. So it'll tell you what the purpose of that panel is. It lists not just the antibodies, but the clones, where they've come from, and the fluorochromes that they're attached to, the types of sample that the authors have used. Most of these are human and mouse and occasionally other primates. There any similarities to any other published OMIPs? Importantly, a figure. You know, we often talk about the importance of publishing data that we show a gating tree, a gating scheme, so that we know how people have derived a particular population, a particular number. And this only takes a couple of pages in the journal, but there's also a whole pile of supplementary online information, which not only includes the data files, so you can download those and play with them yourself, but it tells you something about the way that the authors have come to that panel. So maybe some of the antibodies or fluorochromes that they didn't use, and why didn't they use them? Some idea about the titration, and some idea about that journey we talked about earlier, that testing and iterating that we all have to do in multicolor analysis. So I wanted just to give you a quick example. So this is OMIP 16. Actually, we've just published, I think, OMIP 81. So there's quite a lot of them out there. And this was looking for antigen responsive T cells in humans and monkeys. So you can download this and have a look as well. This is of necessity going to be a very quick overview. But as we've said, they give you an overview, you know, why this panel was developed, because they wanted to look for those antigen specific CD4, CD8 T cells. They wanted to do it in the macaque and in humans, and also in fresh and frozen lymphocytes, because often we might see a difference in the expression level of some of those markers. And it's similar to another couple of OMIPs out there. This is a very useful table, very important table in all of the OMIPs. It tells you which marker they've looked for. So there's a whole series there. It's only a 10-color panel, not a very huge panel. There are OMIPs out there that cover 40+ different markers now. It tells us the fluorochromes that they're using, the clone that we're using, and why we're using them. So CD3, CD4, CD8 are defining markers, they're lineage markers. We have the functional markers here, and then we have activation and differentiation marker. Now, not every marker in a panel is going to be equally important, and this is one of the important things, I think, in panel design. So the OMIP covers that. So we can rank the importance of the markers. So the CD3, CD4, the CD8, the lineage markers, they're fairly low down the pecking order, because we know that they're positive or negative, they're on or they're off. So we may not need to worry too much about the fluorochrome that we're using there. But the functional markers, they're the ones that are the whole point of the assay. So they're the ones that are going to assume the most importance. Now, the OMIP itself will include a description of the cytometer that's been used. So this particular picture is not in the OMIP, but it's one that I quite often draw out for people so that they can help. It helps understand where these particular markers are being added. So this shows that they actually had at their disposal, remember this is quite an old OMIP, they had a 4-laser cytometer, UV, violet, blue, red, and the little white boxes represent the filter spaces that they have in there. And 10 color, they had 13 colors available, so it's not quite maxing out, but not too far off. And this shows the fluorochromes, the positions in the spectrum where they've chosen to put each of those markers. So you could look at that and you can critique that, or you could use that, in fact, if you had a similar cytometer, or if you have a cytometer now with more colors from a violet laser, yellow laser, maybe you could even improve on that. But it's a good starting point. And as I said, one of the other important things is the supplementary information. So you can get information about all the titrations that they've run and why or why not they've chosen a particular marker. If I just go back one slide, the important markers are interferon gamma, the CD154, the IL-2, and the TNF alpha. Now, 3 of those have been put on the first detection channel in the laser, and as we know, that's the channel that's probably going to get the least interference, so the least number of other photons ending up in that particular channel. but one of those has had to go on here, on Alexa 700, which you might be aware is not a particularly bright fluorochrome. But in the supplementary figure, you can see that they've actually tried that TNF in at least 3 different markers. And although PSI7 is a relatively bright marker, actually we don't get as good resolution of this population as we do with APC or Alexa 700. So, that gives a rationale to why they've chosen Alexa 700. And we also get a gating tree, as we saw earlier from what Kathy was doing, single cells, scatter gating, live cell gating, and then looking at the CD3s and either the CD4 or the CD8 population, and then the expression of those functional markers. So, you may never do anything this particular type of biology, but sometimes these papers are actually quite a good thing to look at, because it does help you understand how that panel has been developed and how that fits into the cytometer that it's been run on. Remember, those 2 things are equally important in panel design: the reagents, the fluorochromes you're using, the cytometer you're using, and the biology that's underpinning all of that. So I was just seeing if there's any final questions. Now is your chance, if you have any final questions, to put those into the Q&A or the chat. I don't know if you've got any final remarks, Rui or Kathy?
No, I just want to say that, well, I want to thank our scientific director, Anna Longini, for providing a lot of the data that we showed today, including the FMO. So I forgot to mention that. So thank you, Anna. And I don't know, I think Kathy had to run.
Yeah, I think she's got a sort going.
Right. So,
yeah.
So I might say goodbye for her also.
Yes. So we'll just have a couple of wrap-up slides. This, as I said, this is being recorded this webinar, so it will end up on the OpenFlow Cytometry YouTube site probably early next week. And if you subscribe to that, obviously you'll get a note when that appears. But if you don't, well, why not go there and subscribe?
Right.
And we're going to take a little bit of a break now with these. We thought December will give you all a bit of time off, and we're going to come back in the new year, and we've already got a date booked for our next OpenFlow session, which is January the 14th, 2022. And we're going to look at DNA analysis by flow cytometry. Now, even if you never do DNA analysis, we're going back to doing some live analysis here. We'll be running samples on one of our cytometers. And there will be a lot of tips about how we set the machine up, how we prepare our samples, how we do our data analysis. So, even if you don't do DNA analysis, there'll still be something there for you. And of course, you get to see us again, which is always a bonus. The registration for this is already open. If you don't get the chance to write that URL down, you can just go to the What's On page at the CRIC, and you'll be able to find that. Or you can contact us. The best way to contact us is via the OpenFlow Cytometry Gmail address, which comes to all of us, although our individual email addresses are there as well. And of course, if you follow us on Twitter, you'll get information about everything that we're doing, stuff that we put onto the website, upcoming webinars, and so on. So as usual, we've slightly run over time, but not too bad. So thank you all for sticking with us.
Yeah, but I would just add that if you guys want to add comments to the, to the videos once they're up, feel free. If you have questions, we can also answer them on YouTube. And it's actually nice because then there's a discussion, and that actually can bring more questions in. So feel free to use the OpenFlow YouTube channel to comment on the videos.
Exactly, and some of our earlier ones have had quite a little bit of discussion going on. So all those comments are there, and they're freely available to everybody. Right. So exactly. Yeah, please get in touch with us.
Yes. Okay.
Okay. So thanks very much, everybody. We won't see you for 2020. So however you're going to celebrate the next few weeks and the holidays, have a good time, stay safe, and we'll see you next year.
Yes. Goodbye.
Bye all.